Okay, hello everybody. I can see we've still got a few more people joining, so we're just going to give it 1 or 2 more minutes. But first off, thank you all for joining us here today. All right, So this webinar's going to be hosted by BPES, and we're supported by our suppliers, Constant Systems, who are based in the UK, in Devonshire, and we've also got Lindsay McDermott from the University of Bedfordshire. So we're going to be giving a bit of information on ourselves as BPS, and we're quickly going to jump into Constant Systems and their high pressure cell disruption technology. And the bulk of this webinar is going to be Lindsay McDermott's research on the ZAG protein and its use in obesity and cachexia. All right, we shall begin. So BPS, we're based in Kent in the UK, so we're down in the southeast. We provide scientific equipment, process equipment used in cell and gene therapy. Um, our business is broken into three main sectors. So it's cell line development work, upstream processing, and downstream processing. So we sell everything from bioreactors to cell disruptors, centrifuges, TFF systems, quadraflow pumps, and everything in between. So this slide here gives you a bit of an overview of the main industries we support. Along with supplying the equipment, we also have full service and support. So we install the equipment, we provide training, and we keep that equipment running throughout its whole lifetime. So there's two halves to our business. Here's just a little overview of our main distributors, so the companies we support and supply for. In a lot of cases, we have exclusive relationships with these. So if you're wanting to buy equipment through these companies, it goes through us and we will support it and you throughout your work. And today we're going to be focusing on Constant Systems. As I said, they manufacture high pressure cell disruptors from small benchtop scale all the way up to full flow production. So with that, I would like to hand over to Jay, and he's going to tell you a little bit more about Constant Systems. Thank you.
Many thanks, Matt. Hello, everybody, and thank you for joining this webinar. We have the pleasure of Dr. Lindsay McDermott speaking to us about lipid binding by zinc alpha glycoprotein, ZAG, towards a treatment for obesity and cachexia. It's not an easy word to say. Dr. McDermott is from the University of Bedfordshire, where she has a state-of-the-art laboratory, and her research is receiving a lot of attention. My name is Jay Champaneri and work for Constant Systems, partnering BPS as our distributors in the UK. I don't want to take too much time from Lindsay's presentation, so in short, I'd like to highlight the essential differences in technique our platform has to offer relative to other methods for cellular lysis. So in the laboratory, we're all familiar with sonication, homogenization, chemical lysis, whether this be for microbial cells, mammalian cells, plant or animal tissues, fresh or frozen. But these techniques have their place too. The cell disruption technique using the Constant System platform has been widely used, and there are over 3,000 publications citing the use of this technique to generate process samples efficiently. We use orifice cell disruption for the entire sample, which experiences the same process, even at scale-up. Once the optimal parameters are set, the process remains the same, day in, day out, person to person, laboratory to laboratory, country to country, with reproducibility being of paramount importance between collaborating laboratories. All this with ensuring the highest yield from a single pass and sample integrity due to the platform's unique cooling systems. So as we listen to Lindsay's presentation, please think about how your sample is best processed for your application for downstream analytics. This slide actually shows you the range of products that we have. So in terms of being able to scale up, we have the ability to take you from small scale sonication devices and homogenizers all the way through to higher throughput processing. But I'm very much a visual kind of guy. So if you have applications where you're utilizing yeast, E. coli, mammalian or insect cells, then these systems are used widely for these applications. We're going to dwell much more on protein crystallography with Lindsay's work, but I just wanted to give you an oversight on what capability Constant Systems has. The nice thing is we have one technology, and we believe that we do this really well throughout the whole scale-up process. I'd like to show you a video on how the system is used in Lindsay's laboratory, and it shows a process that is very simple but effective. Just press that. And with that, I'd like to hand on to Lindsay for her presentation on this really exciting piece of work.
Oh, thank you, Jay. I'm just going to share my screen, and hopefully everyone can see my screen. So first of all, I'd just like to thank BPES and also Constant Systems for inviting me to give this seminar. My seminar today is lipid binding by zinc alpha-2 glycoprotein, or ZAG, towards a treatment for obesity and cachexia. And in this work, I'm going to be featuring I'm going to be describing how we have used the Constant Systems CF1 cell disruptor. Okay, so to start off with, I guess we're all familiar with obesity. As humans, if we eat or take in more calories than we expend the energy for, we end up depositing fat in our bodies. So obesity is a metabolic disease, and it's categorized by body mass index. So the higher our body mass index, different values of BMI are used to categorize different levels of obesity. If a patient is classified as being obese, they are at higher instance of experiencing cancer and also inflammatory diseases such as type 2 diabetes. Perhaps lesser well known than obesity is cachexia. So cachexia is a metabolic syndrome. It's a syndrome because its cause isn't known. And cachexia is a loss of muscle and fat tissue. It doesn't matter how much you eat, you never ever can regain the muscle and fat tissue that you've lost, and eventually patients suffering with cachexia die of it. So cachexia is a syndrome that accompanies cancer, and it's often responsible for the death of cancer patients rather than cancer itself. And it also affects people suffering with inflammatory diseases, so things like chronic obstructive pulmonary disorder, kidney disease, heart disease. It also affects the elderly, healthy elderly, and causes frailty in them. Zinc alpha-2 glycoprotein, or ZAG, is a protein that is associated with both these conditions, obesity and cachexia. And what you're seeing on the screen here is the structure of the ZAG protein. So the ZAG protein is shown in red and blue. So ZAG, if we were to describe its protein fold, it has a class 1 MHC protein fold, where MHC stands for multi-histocompatibility complex. Now, typically, class 1 MHC proteins are involved in the immune response. ZAG, as far as we can tell, isn't involved in the immune response. And we are interested in investigating how lipids bind to this protein. So the ZAG protein shown in blue and red, and the lipid is shown in the middle here in green. Now, this protein is made up of 3 different domains. We have an alpha 3 domain down the bottom, if you follow my laser pointer, And up the top here, we have two domains, alpha-1 and alpha-2, that join together to form this groove region, which is where the fatty acid shown in green binds. Okay, so normally these proteins, these class 1 MHC proteins that look like this, they're normally involved in the immune response, and normally in this groove they bind peptides that get presented to antigen-presenting T cells, and they induce an immune response. As far as we can tell, although ZAG looks like it should be involved in the immune response, it's not doing anything to do with the immune response. So what can I tell you? Where do we find ZAG in our body? Well, it's expressed in a whole range of different tissues, and it gets secreted into our plasma. It also gets secreted into saliva, into prostatic fluid, into sweat. We, of course, are interested in obesity and cachexia, so I can also tell you that ZAG is expressed in fat cells. So here, if you follow my laser pointer, this is a fat cell. The fat cell is filled with this large white area that is triglyceride or fat. So ZAG is made from fat cells and it gets secreted from them out into plasma. If you look at the levels of ZAG that are found in fat cells or adipocytes from obese patients, these levels are reduced in comparison to weight-stable controls. And the opposite is true if you look at the amounts of ZAG that are found in the adipocytes from cachectic patients. So here, levels of ZAG in fat cells are increased compared to those from weight-stable controls. So what is ZAG actually doing? Well, scientists can tell you, they know that if you take ZAG and you incubate it with fat cells, it causes the triglyceride or the fat that's stored inside the fat cell to be broken down into its sort of building block molecules of glycerol and fatty acids. It's also known that if you take white fat cells like this one and you incubate them with ZAG, you can turn them into brown fat cells. So brown fat cells are shown over here on the right. They're filled with mitochondria, and it's the mitochondria, the energy source of the cell, that produces the brown colour. They're also filled with much smaller lipid droplets. So the big question really is, how does ZAG do this? How does it communicate from outside the cell to cause the breakdown of fat inside the cell and to cause the conversion of white fat cells into brown fat cells, these brown energy burning fat cells? So that's really my research aim. I want to uncover the molecular mechanism by which ZAG participates in lipolysis and the browning of white adipocytes. If we can be successful in doing this, it could potentially provide the NHS with a new, more cost-effective strategy for treating obesity, type 2 diabetes, and cachexia. So our research questions, ultimately, we're at the very, very beginning of this process, and we want to know, what is it that ZAG binds to in the body? What are its physiological ligands? So far we know that ZAG contains one loose fatty acid binding site. We also discovered that it contains 15 loose zinc binding sites all over the structure of ZAG. And while we were investigating those loose zinc binding sites, through the experiments we were doing, it led us to believe that actually the ZAG groove contained 2 distinct fatty acid binding sites rather than just one. So this wasn't a crazy suggestion. So here's our structure of ZAG. We already knew from experiments that fatty acids bound to ZAG in its groove area here. There was also another protein though that's found in humans called CD1C. It's also an MHC-like protein. It has a very similar structure to ZAG, but it is involved in the immune response, and it binds bacterial lipids and it protects us from infection with bacteria. So here you can see this protein is binding also a fatty acid in green, but it also has this large molecule in yellow. That's a phospholipid. So we potentially wondered if something similar was happening with ZAG. Now, a couple of years ago, we crystallized ZAG with a fatty acid. So the fatty acid that we used in these experiments was called DAUDA. So this is DAUDA shown here. If you follow my laser pointer, it's a C10 fatty acid, and attached to one end of it is a dancile fluorescent group. So when we made a complex of ZAG with this fatty acid, we were lucky enough to crystallize it. We got these crystals. We took these crystals, we put them in front of an X-ray beam, and we collected an X-ray diffraction pattern. And from that X-ray diffraction pattern, we're able to work backwards and work out the coordinates of every single atom in that protein to build up its three-dimensional structure. Now, we were expecting that we would find the structure to look like this, with the fatty acid bound in this, with the DOUDA fatty acid bound in this green position here. However, that's not what we saw. When we solved the structure, this is what we saw. So what we actually saw was that ZAG was forming tetramers. So tetramer just means 4 molecules joined together. So if you follow my laser pointer, here we have 1 ZAG here, 2, 3, and 4. And these are all joining together to form this tetramer. The tetramer is formed by overlap of the alpha-3 domain. So up here, if you follow my laser pointer, is the A1, A2 groove, and down the bottom is the A3 domain. So we had 4 A3 domains overlapping to form what's called a beta barrel structure. It's almost like a kind of pore. So first of all, we were flabbergasted that we saw these tetramers, but also what we weren't expecting was that we also saw the fatty acid DOUDA, which is shown in black here. We also saw it bound inside these beta barrels and associated with the alpha-3 domain. So this is something we weren't expecting at all. The other thing that we weren't expecting was that in the alpha-1, alpha-2 groove up the top here, we also saw not just 1 DOUDA molecule binding, but in some cases up to 3. So from this finding, we thought this is potentially relevant to this disease state of cachexia. So cachexia, where we're, if you like, undergoing lipolysis, releasing a lot of fatty acids. Those fatty acids are not soluble in our blood plasma and they need to be solubilized somehow. So if we have high levels of ZAG protein and high levels of free fatty acids, then this potentially might act as a mechanism of transporting fatty acids away from adipocytes in cachectic patients. Around about the same time as this, or following on from this, we also were exploring in much more detail what was happening in the alpha-1, alpha-2 groove. So here is monomer ZAG protein. This area highlighted in blue is our DOUDA binding site that we know all the amino acids that are involved in this, not only from the crystal structure, but also from other site-directed mutagenesis experiments. So from some experiments that we'd done when we were looking at zinc binding, we were led to believe that another fluorescent fatty acid called C16-BODIPY, and shown down here at the bottom of the screen. So it has 16 carbons in its fatty acid chain and it has a different fluorescent head group. We thought it was potentially binding further down the groove down here. So to test this, we made mutants. So you can see if you look at this area over here, it highlights all of the amino acids that we thought were potentially involved in interacting with this second fatty acid. We mutated each of these residues to alanine, and then we expressed those proteins, purified them, and we compared their structure and their function in comparison to the wild type unmutated protein. And without going into lots of detail with you, what we found was that this amino acid Y12A in the groove, when we mutated it, it caused the protein to misfold. We also found that the amino acids A158 and R157 and K147, when we mutated those, we disrupted the C16-BODIPY binding ability. So what that told us was that the groove as a whole, this other part, this lower part of the groove, was also active in lipid binding. And so potentially that the ZAG groove might be able to bind bigger and larger lipids than just fatty acids. In all of this work that we have been doing, we have been using recombinant E. coli ZAG. So we need to express and purify our ZAG protein from E. coli. We then use this protein in biophysical studies and structural studies to learn more about how the ligand or lipid is interacting with us. So as a reminder, in order to be able to produce recombinant protein, we need to start, first of all, with vector DNA. So here is vector DNA. Into that vector DNA, we need to insert our gene of interest. In our case, that is ZAG, that's shown here in blue. That vector also contains an antibiotic-resistant gene. Once we've made our clone, we insert that piece of DNA into our bacterial cell in a process called transformation. We can then grow those bacteria on a plate containing the antibiotic that is in our gene clone, and that allows us to select bacteria that contain our cloned DNA. We can then take that bacteria, we can use it to inoculate large volumes of media also containing antibiotics, and that allows us to amplify or grow grow the bacteria and create many copies of the cloned DNA. At a certain point, we induce these bacteria by adding in a chemical called IPTG, and that causes the bacteria to start making or transcribing the protein, transcribing and translating the protein. We leave that to incubate for a while, and then we collect all of the bacteria that are now filled with the ZAG protein by centrifugation. We then take those the bacteria and we need to lyse them or split them open, and more on that later. This is where the CF1 cell disruptor comes in. But typically after you've lysed or broken open your bacteria, if you centrifuge it, you'll receive a pellet of insoluble material, and all the soluble proteins would be found in the supernatant. You then take the supernatant, which contains a mixture, your protein of interest, along with a mixture of other bacterial proteins, and you pass it down various protein purification columns so that you purify out your target protein that you are interested in. You can take elutions off of these columns and run them on SDS Page to analyze the size and the purity of each of the samples that you've collected. So for us, when we are expressing ZAG protein, unfortunately when we express it in bacteria, it's not soluble. Instead, it forms inclusion bodies. So inclusion bodies are misfolded protein, basically made up of large protein aggregates, and they are insoluble. So what this means is when you lyse your cells and spin them, your pellet contains not only insoluble materials such as cell membranes, but it also includes inclusion bodies. And in the supernatant would be other soluble bacterial proteins. So for the last couple of years, we have been running at the University of Bedfordshire a training event in collaboration with the Biochemical Society to train people in the use of protein purification and various aspects of biophysical characterization. And it was at one of these events that we were introduced to Constant Systems, and we started discussing the work that we were doing. They suggested that we might be interested in using one of their instruments, the CF1 cell disruptor, as part of our cell lysis. And so here you're seeing a CF1 cell disruptor instrument. On the right hand side, we have Mark Partoon from Constant Systems, and also Sophie Osborne, our technician. So the first thing we wanted to know was, so this, I should say, the CF1 cell disruptor, it works on pressure or high pressure to break open the bacterial cells. So the first thing we wanted to know was, what is the pressure that we need to use to be able to maximally recover our inclusion bodies from E. coli? After that, we then wanted to know, are there any differences in the ZAG protein yield that we get and the protein characteristics compared between chemical lysis, that's the approach we typically use normally, and this high pressure lysis method from the CF1 cell disruptor. So what we did is we grew up 2.5 litres of our E. coli expressing our ZAG protein, we induced the bacterial cultures, we left them for 3 hours, and then we collected all the pellets. We redissolved the pellets in 120 mils of buffer, and then we split that pellet suspension into 5 different samples. And for the first sample, we underwent chemical lysis. So we processed our sample as usual, chemically lysed it, centrifuged it, and we saw at the bottom of our tube our ZAG protein inclusion bodies as expected. We then passed the rest of these samples through the CF1 cell disruptor, and we tried various different pressures. So you can see here 10 kpsi, 12.5, 15 and 17.5 kpsi. So if you look at these, again, we saw when we centrifuged our process samples, we see ZAG inclusion bodies. The 10 kpsi sample was almost equivalent in size, just by looking at I, to the pellets we obtained using chemical lysis. And what we noticed was when we went to higher pressures, the inclusion body pellets were reducing in size. So we knew at this stage that lower pressures, 10 kpsi, was going to be much more optimal for us. So we repeated this again, but this time we went to lower pressures. We went from 10 kpsi down to 5 kpsi. Again, we had our similar control where we chemically lysed our cells. The lower pressures, when we went from 10 down to 7.5 down to 5, we didn't see any difference visually when we're looking at it with our eyes in the size of the inclusion bodies. So we definitely knew that this lower pressure range was a good range for us to be working in. What intrigued us at this stage, though, was that we were losing inclusion bodies at the high pressures. And when we looked in the literature, high pressure was actually a method that is used to solubilize inclusion bodies, to turn them from this insoluble state into a soluble state. So we wondered if that was perhaps something that was happening in our experiments. So to test this, we basically took the supernatants from our samples that had been processed at different pressures using the CF1 cell disruptor, and we ran equivalent amounts, equivalent volumes down an STS page gel. So STS page gel separating proteins based on their size. If you follow my arrow down here, every single one of these bands is a protein of different size, and the sizes are presented in the ladders at either side. Now, had we been seeing solubilization of ZAG at higher pressures, we would have expected to see more bands or higher density bands of 32 kilodaltons in our STS page at the higher pressures. So ZAG is 32 kilodaltons in size. So we would expect to see ZAG round about where my laser pointer is. So if you look at this gel, we don't see any difference between any of these samples across our range of pressures. So we didn't think at this stage what we were seeing was solubilization of our ZAG protein. We also measured the protein concentration in the supernatant assay using the BCA assay. And again, if we were seeing protein solubilization, we would have expected the high pressures to have had the highest total protein concentration in the supernatant, and we didn't see that. So we're not really sure how to explain the loss of our inclusion bodies at this stage. It is possible that the higher pressures are just completely disintegrating the proteins into its individual amino acids. But one caveat for this is what we haven't monitored as part of this process is the extent of lysis. We're assuming we've got 100% cell lysis at all pressures, which we're pretty sure we have for bacteria, which are relatively easy to break open. Right, okay, so we've established at this point that lower pressures are better for our recovery of inclusion bodies. So we took our inclusion bodies that we had created, we refolded them, and then we passed them down our gel filtration column. So a gel filtration column, also known as a size exclusion or SEC column. So we passed our protein down the column, and you can see these are the samples that eluted off. So we ran a sample, our ZAG sample, that was created in red from chemical lysis, in blue from a sample that was created using 10 kpsi of pressure, and in black the sample created from using 5 kpsi of pressure. You can see that each of these samples elutes off the column at approximately 70 milliliters, but the elution volumes are all slightly different. Each of these samples was then run on an SDS page gel. You can see over here, we saw single bands for every single sample. So this is chemical lysis, the sample prepared from 5 kpsi and the sample prepared from 10 kpsi. So we see single bands, pure protein of the correct size. When we calculated the yield from each of these preparations, so we were calculating a total protein yield from 500 mils of culture. For chemical lysis, we got 0.6 mg of protein. From 5 kpsi, we got 0.61 mg of protein, and from 10 kpsi, we got 0.59 mg. So very, very similar yields. We took our sample just to check our sample was actually our sample. We ran it on mass spectrometry. So mass spectrometry gives you an accurate mass for your protein. So based on our amino acid sequence for recombinant E. coli ZAG protein, our molecular weight should be 32.27 Daltons. And you can see when we ran it on chemical lysis, we got a mass of 32, or when we prepared our sample using chemical lysis, we got 32.28 kilodaltons. And when we ran it, when we ran the sample prepared using from 5 kpsi pressure, we got 32.28 kilodaltons. So we have ZAG protein there. Interestingly, in our chemical lysis sample, we had an additional peak at 32.55, which we didn't see in our 5 kpsi lysis. I'm unable to explain why this is there at this stage. It shouldn't be there. The difference in size is approximately equivalent to 2 amino acids, but there are 2 amino acids that just shouldn't ever be there in that protein. So this is something we'll be going back to look at. The other thing we checked was from each of the 3 proteins that we've made, we checked their protein stability. So we can tell how stable a protein is by following how it is affected by heat. So what you're seeing here is, so we've got our 3 different proteins, our protein prepared from chemical lysis, our protein prepared from 5 kpsi and 10 kpsi using the CF1 cell disruptor. And what we're following here is the intrinsic fluorescence of the protein. So that's on the y-axis up here, and on the x-axis we have temperature. So we're slowly increasing the temperature, and as we increase the temperature, you can see we get a drop in fluorescence. The fluorescence then starts to increase as the protein begins to unfold, and then at a certain temperature up here, the protein is fully unfolded. So the midpoint of this transition gives us an estimate of what's called the TM, or melting point of the protein. To get an accurate value for that, we take the first derivative of this data, and when we do that, we get this graph underneath with a single peak. The point in that peak gives us the accurate TM value, or melting point. So so you can see the traces that we get for each of these protein samples, but here are the actual values that we got. So you can see the unfolding or melting temperature 5 kpsi, it was 65.63 degrees Celsius, and from 10 kpsi, 65.65 degrees Celsius. So these were almost near identical. And if you look at T-on is the temperature at which the protein begins to unfold. For chemical lysis, 56.25. For 5 kpsi, 56.45. And for 10 kpsi, 57.01. So we had a bit more variation in the T-on compared with the TMs. So we are seeing differences, but very, very small differences. So overall, to summarise or conclude, The protein yields that we got from using the CF1 cell disruptor at 5 kpsi and 10 kpsi, those protein yields were very, very similar to those produced from chemical lysis. When we look at the stability of the proteins, we can see that the TMs of the proteins are almost identical. All three proteins have near identical TM values. We are seeing these small differences in the T-on value. From our perspective here, we are happy that the pressure that we would be using in the CF1 cell disruptor would be 5 kpsi. That gives us protein that is comparable, definitely in terms of stability, to our chemical-lysed protein. We are going to go on and do further experiments to assess the secondary and tertiary structures of each of these proteins to see if there's any differences between them. But going forward, we will be using 5 kpsi as our pressure to disrupt our bacteria to produce recombinant E. coli ZAG protein. One thing that I haven't mentioned, obviously we've been going through all of these chemical tests, one thing that I haven't mentioned and that we see as a huge advantage is that the CF1 cell disruptor for us and what we do is a massive time-saving device. So for in our chemical lysis process, when we are lysing our cells to release inclusion bodies, we need to wash our inclusion bodies, and that process it involves various centrifugation steps that are repeated many, many times. That typically takes us, depending on the experience of the user, either half a day or a whole day. With the CF1 cell disruptor, regardless of the experience of the user, we're basically processing these samples within half an hour, if not less. So we're saving almost half a day to a day in process time. Okay, so that brings me to the end. All that I need to do now is to acknowledge the people who were involved in completing the work that I've just told you about. So Sophie Osborne is a graduate of the University of Bedfordshire, both at undergraduate and master's level. She's now working as a technician at the School of Life Sciences. So she is the person who generated the data that I've shown you today using the CF1. Rian Fox, Mark Pertun and Richard Malabar were the people who talked us through and helped us through optimizing the various pressures and helped us understand what we were seeing when we were using the CF1 cell disruptor. Ben Revel from Nanotempter helped us run our stability experiments, and Kirsty Carew from UCL helped us run our mass spectrometry experiments. So thank you for your attention. I'm going to hand back to Matt, who's going to field questions.
Okay, thank you, Lindsay. Really appreciate you taking the time to present that there. I think everybody found it very interesting. Obviously, there's more research to be done there, and we're interested to see where it goes.